Good morning, everyone.
Good morning.
Good morning.
Morning.
No, I think
Good
she hasn't
No,
joined
morning.
yet.
not yet.
Yeah, sure, Louise. Yeah.
Yeah, sure. I don't mind.
Guys, have you guys got all your placement location?
I got mine already.
Me too, I don't have a location yet.
I got mine,
Oh,
but it wasn't a preferred one. But then I said okay to it because the ones that I wanted to go for, they were not available.
me too,
Yeah,
but
yours is
there's
Niagara.
no.
Mine's in like Brampton, but closer to Saga. Brampton is the Interpret group of medical.
But you're closer to Brampton, right, Shabana?
No, I'm actually in the heart of Mississauga. I'm like near
Oh,
Square One area.
I see.
Yeah, I wanted the Interpret group in Mississauga, but they had the Interpret group in Mississauga. It's not available apparently. But then the one in Brampton they had available. But I wanted to go with that medical group.
Anyway, you're driving, right? So it's
Yeah, I mean
just
it's
fine.
if
Yeah.
it's no traffic, then it's about I saw the thing was around 14 to 15 minutes drive, which is not too bad. But with traffic, like around 5 p.m. and stuff, it's a long drive. It shows
Oh
like
yeah, that's
35
only the
minutes.
problem in the afternoon because it's rush hour.
Yeah, so rush hour
Yeah.
shows like a long drive, but then if it's not rush hour, it's not too bad. It's 15 to 17 minutes.
Mine, I haven't heard from the set yet because Oakville, we only have a few locations. So,
Yeah, yours is Oakville,
yeah,
right? Like I want in Saga. She's just like, it's not necessary you'll get in Saga, but maybe close enough. I said, okay.
I might gonna talk to her on
Yeah,
next
but I'm
week.
sure she'll
Yeah.
get it for everyone because there's still some time. So I'm sure
Yeah,
she'll make sure that everybody gets a placement.
right. Yep.
Do you guys want to go through the questions while we're waiting for Miss Munman? Okay, no problem. I'm trying to call her. Actually, she didn't pick up, you know. It says calling, but then it just got dropped after a while. Oh, she just sent a message in the WhatsApp group, actually. No, she said that she had internet problem. Yeah, she just replied. She's just like had internet problems, so I think she'll join soon enough. But if you guys want to go through it together, just let me know, okay? I'm also just working on the questions.
Yeah, me too, Shabana. I'm still working on mine,
Yeah,
but
I have
maybe
the
later
last few.
on.
Yeah, I have the last few pages left, but then, you know, like if everyone wants to just do it, then
I
let me
think
know.
she
Whenever you
wants
guys are
us,
ready. Whenever you
I,
guys are ready, no worries.
I think she wants us to read, continue to read the the slides.
Yeah.
I think that's what she mentioned yesterday.
Oh, she's on. She's on now.
Okay, good.
Okay.
Sorry, dear. My internet was not working. My goodness. That's why I cannot do anything.
That's
Oh,
okay, ma'am. It's fine.
I have to
Yeah, it's okay, miss. I had the
yeah.
same issue logging on this morning too. Like my
Oh my
internet,
God.
I don't know.
Good morning, ma'am.
I was joining, it's not allowing me to join. And then nothing WhatsApp also I cannot communicate with guys. The internet was not problem.
Yeah, no, it
Yeah,
does that
that's okay,
sometimes.
miss.
No, I
Yeah.
know it's okay, but it's give just the stress. That's the thing.
Yeah.
It give you
Unnecessary
this
stress.
unnecessary stress. You give the
Yeah,
stress. Yeah.
you're right.
Yeah. So let me just go quickly. Uh, Files. Okay, so editing, opening Excel. Where we are now? Control, page. Two, two. Microbiology. 2 unit 6. We did the unit 6 and now.
Hi
All
guys.
set.
Just sorry, just a quick question. Uh, is there any information I missed on WhatsApp? Because my tutorials no any information from my WhatsApp, so I just want to know if I missed something.
No, Ying Hong, there's nothing new on WhatsApp. I don't think you missed anything.
Okay. I don't know why my WhatsApp. What happens to my WhatsApp? Thank you guys.
No problem. If there's anything, I'll message you on Teams then, okay? I'll let you know.
Oh, thank you, Shibana.
No worries.
Okay, so we are going to unit 6. Uh, what is that? Okay. Share this one. So far we have learned about what is microorganism, how to identify them, using which instrument, which stain, how we can culture them, grow them, how we can culture, what are the different mediums, sources, how we can culture them so that we can recognize or identify them. Now today we are going to learn about how we are going to the process of actual, how we are going to culture them. So we do inoculation and incubation for bacterial growth. So here we are going to mainly recognize the different cultural media required to inoculate plate based on microbiology specimen type. Different specimen needs to be inoculated and cultured in different way. Discuss methods to use to identify bacteria. We already did. Why they are going to repeat it here, I don't know. We did gram stain culture, supportive test. Describe the procedure for inoculating agar plates. This we are going to learn how we are going to inoculate the agar plate. How means how we are going to put the bacteria in the agar plate, in the slant tubes and broth cultures. Explain the purpose of isolating colonies. Why you need to isolate the colonies, which is giving you individual kind of bacteria, and also various types of incubation requirements needed based on the specimen type, media, as per the type of bacterial infection. So this is the bacterial cultivation. We all know cultivation means grow. So the process of identification of microorganisms begins with inoculating various nutrient media designed to enhance the growth of pathogenic organism. So cultivation means you have to enhance the growth of pathogenic organisms using different types of nutrient media, whether it is agar plate or in the broth, in the slant culture, but all these media has nutrients. What nutrients? The nutrients which is helpful to grow the bacteria. And also you have learned yesterday that different selective media. What is the difference between selective media? What is the enriched media? What is the differential media? So also it depends upon what sample or organism we are culturing. And then what type of stain you can use to identify them. So straight plate method previously described used to separate. It didn't describe previously, it's a wrong one. Used to describe separate organisms. Oh dear, what is happening? Can you hear me? Hello?
Yes,
Hello?
ma'am.
Yes,
Yes,
ma'am.
ma'am, we can
Okay.
hear you.
Yes, yes.
Suddenly something happened in the computer again. So, oh yeah. The straight plate method previously described is used to separate organisms to obtain a pure culture that looks of what is I am telling, right? So, how you are spreading in the agar plate the bacteria to get a pure culture. That's we are going to show you. Once a pure isolate is identified or defined, the organisms can be utilized in the biochemical test for identification purposes using the different type of stains. In addition, pure culture is used to test for susceptibility and resistance to antimicrobial agents to check which one is effective.
Miss, I'm not
This
hearing you.
again, everybody.
We hear you,
No, I'm
ma'am.
good.
I
We
can
can
hear
hear
can
you.
hear
you.
I
you.
can hear you.
I am
We can
really
hear you.
Asnet, you cannot hear me. Others can hear me?
Yes, we can hear you,
Yes,
miss.
ma'am.
Yes, we can hear.
Okay. So, okay. So now we have to look. For doing that, we need a couple of instruments and basically the techniques. Microbiologists use special equipment and techniques to grow and isolate pure cultures from microorganisms. Most work in the microbiology laboratory involves procedures, same thing they are telling, to culture, to characterize, and to identify various microbes. So, what you are going to learn here, culture of organisms present in the patient specimens, classification and identification of the isolated organisms, whether it is from one specific type of bacteria or is a mix of bacteria or kind of these things. So what type of infection the patient has, and the interpretation of organism susceptibility patterns to determine the use of an appropriate antimicrobial agent. So that means doctors can, from their results, doctor can understand what type of treatment method doctor has to give the patient. Then what is that? Such efforts require the use of specific techniques. Is we know that, yes. So we need inoculating needle or loop. You cannot do by hand. You cannot do by. So what you are going to do? There are some inoculating transfer needle or loop to spread the organism onto the agar plate into an artificial nutrient growth media. What is called artificial nutrient growth media? Because you are making it, you are putting them what you made it or you have it in the lab. So that is the artificial nutrient media which is ended with everything what the bacteria or any microbes need to grow. Needles and loops may be disposable or reusable. So this all has to be sterile 100%, so that no contamination should come from them. So either use and throw the inoculation or bearer loops which you can autoclave or reuse. Disposable inoculating loops are calibrated to deliver accurate volumes, and liquid dispense are made of plastic and can be discarded after use. What I told, reusable loops or needles are typically made from platinum or an alloy such as nichrome that can be sterilized by heated to glowing without being damaged, that returns to room temperature fairly rapidly. So it's not iron. So this is the thing, that needles. What about the inoculating loops? You are going to see the picture layer is made up in such a metal. That is called the platinum, or their alloy is called nichrome. How you sterilize them? You put in the fire. Actually, you can see there is a, so you put the heat and it's going to be red. That way you can sterilize it, and then but when you can take it out and everything is going to be the under biological safety cabinet. When you take it out, it's going to be return to the room temperature rapidly, and then you can use that sterilized loop for the inoculation. So the metal needle loop is used to distribute specimen and transfer microorganism from one medium to another, from a culture to a microscope slide. So from one culture to another culture, even onto the microscope slide. The needle or loop can be safely heated. It is sterilized almost instantly with an incinerator or Bunsen burner. So on the direct flame or incinerator, which is give you the, it's not, Bunsen burner is generally give you the flame, but this one is like the hot, something is going to, you can see there is hole and everything is going to give kind of a flame, but the hot, like you have the stove, you can see the It's kind of inside the coil things, and you can see the fire, hot red things. That is like that. Because it can be sterilized quickly, the loop can be used repeatedly for this purpose. Before placing the needle or loop into the specimen of the media, the needle or loop is sterilized and incinerator or flame. That is their telling for doing. And for between each one, you have to, so you are using one plate, then again you have to sterilize it with the incinerator, and then again, so always you have to sterilize it back and forth, back and forth. So this is the loop, and this is called the incinerator. You are going to see it in our lab. This is the electrical plug, and inside that loop is hot. You can see the red color. So it's so hot. And this is the incinerator. This is the loop. You are going to put the loop inside that one. And you can see the loop is also getting red. And then once that is totally red, then you can take it out. And this is the Bunsen burner where you have the direct flame. There is also you can put the needle. But nowadays, due to the risk, generally people doesn't use this one anymore. We used to use this one. So these are the different types of inoculating loops. This what that loops means. It has a handle in the back. This portion is made out of that platinum or the special lithium metal, which is extremely heat tolerable and can come back to cold very quickly. And what is called the loop? You can see in the front there is a loop, like a spoon, very thin spoon with a hole. So these are the loops. This is the incinerator. So you are going to put this loop inside this one. You plug it, it's going to be red hot inside. And this is the Bunsen burner where you can get the, from the gas, you have to get it and you can get the flame. So these are the plastic loops, which is use and throw. So it comes either sterile. There are different types of loops. Mainly people use this kind of loop. This is called the hockey stick loop. I never use this one, but there are different kind of loops regarding depending upon their holes or no holes, depending upon how much you want to inoculate. So these are called the needle or probe of the loop. Choosing the correct media, solid plates, slant or liquid. So if you are going to culture the CSF fluid or for the checking for the TB, you have to use the specific media. When you are doing some fermenter from the, so it depends upon what type of requisition, what type of problem or the patient has and what type of culture you need. For the basic culture for everything, sheep blood agar is good. But sometimes for getting the good growth, sometimes you may have to select the specific culture media or plates. The necessary media for culturing is dependent on the source of the specimen and what type of culture and sensitivity is ordered. For the urine, what are you going to use? For the CSF, what are you going to use? For the stool, what are you going to use? That we have learned yesterday. For example, a throat specimen is plated on different plates than a urine specimen. The type of incubation is different as well as based on the pathogenic bacteria typically found in those source infections. Right? Urine will have different type of infection than the throat. In throat, we have more staphylococcus, throat infection. The urine, we have more the UTI related. So see the chart on the next slide and identify the plates and incubation requirements for those two specimens. So throat, media, is BAP of CNA, incubation 35 degrees centigrade, anaerobic. So they are giving all these things where you are going to do that one. So nasal, McConkie agar. So this is, you can use the BAP. So here is the chart. What type of cultural media you are going to use and what type of stain you are going to use here and what is the incubation temperature. So basically you can remember for the stool, specifically what you can do, what is the Campylobacter, what you can do. So this, like yesterday we have learned, that is very important to remember. There will be a lot of questions on that. Okay, now safety equipment. Obviously, we have to have the work in the biological safety cabinet, right? And work left to right. And you have to work in such a way that you are not blocking the airflow. Because the airflow, the HEPA filters, and the air flow is the main principles which takes the any pathogen or microgen away from the worker. And also it is going to be going through the HEPA filter for depending upon what type of biological safety cabinet are used. Either it is going to go in the air atmosphere or sometimes it is getting purified. There are different types of biological safety cabinets, but in the basic labs we use the biological safety cabinet too. But when you are really working with the different kind of high risk group viruses or bacteria, then you have to use the specific biological safety cabinet. Now, how you are going to plate it? What is the purpose? Purpose for plating in the agar plate to isolate the bacteria present in the specimen is necessary for identification. To do this, all plates are stretched into 4 regions. So you can see 1, 2, 3, 4, 4 regions. So one streak, and then you are taking from here. So here you are going to making the, you are adding your sample here with the loop. Then with the loop you sterilize it in streaking it like that. This is called the streaking. You are spreading. Okay. So then here is the original one. And then from here you are not, now you are diluting because this is going to be too much bacteria there. So then you are going to take one touch, one portion of this one and then streak again this. Leaving this one in the third, again you are going to take from here, you are going to spread like this. And the last fourth, you are going to do like this. So ultimately you can see the bacteria colony, how it grows. So when it grows, heavy growth. This is from you are starting this one. Then you are getting little bit less. They are getting diluted. So you can get individual colony if there is mixed population of bacteria. From here, you are again diluting them and then you are staking like this. So this is the isolated single colonies. So you can see from a huge dense growth of bacteria, you can get the single isolated colony.
Yes.
So it is yes.
So you take from the original and just keep spreading it and it get less and less. Is that what it is?
Yeah. It's
Okay.
not, it is not less the pathogen, but it's you are getting diluted to get the pure culture. Say if it is a picture of diplococci or what is called spirochete, say for example, any infection. So when you are doing this one, you are diluting, and then this one, you can get a colony of diplococci and also spirochete because they are doesn't go in the same time, same, but here they are all together.
Yeah.
Here, they are spreading to get a isolated individual colony.
Okay. Thanks.
Okay. So we are going to see the video how to properly strike a Petri plate for isolated colonies, and you are going to see in the lab, and you are going to do it also. So it says this is a great video by Hardy Diagnostics on the purpose of techniques for the 4 quadrants techniques. Okay, so let me give the hints. If you using a metal loop, insert it between each quadrant. That is called, when you are using the metal loop, you are first insert the loop, strike it. Again, insert the loop, then you strike the second time. So each time you are going to sterilize or insert the loop, the metal one. The plastic loop, you can flip the loop between Q1 and Q2, reduce of bacteria. So generally, what we do is set only Q1, Q2, and Q3 was taking the next quadrant to reduce the number of colonies, whatever. Generally, we never use the same loop. If the plastic loop or the disposable loop, you inoculate, we throw out, we use another one. Or what they, if they have some handle or something that the needle, you can exchange it. But the best one is to use a metal loop. Okay. Okay, so let us see. This is unit 6. In case it goes away, what is the slide number? X16. Okay.
In order to correctly treat an infection or identify a contaminant, microbiologists must first isolate the target microbe from a sample or specimen that often contains other microorganisms. Streaking agar plates is an essential technique used in the laboratory to isolate pure, individual colonies from a larger sample source. Before beginning, it is important to remember individual microbial cells are not visible to the naked eye. Avoid the tendency to over-inoculate the plate, a common mistake that will cause the plate to be overloaded with colonies and result in a confluence of growth. This is particularly critical for certain specimen types, such as stool samples, where isolation of single colonies is extremely important. Also, agar is similar in consistency and firmness to Jell-O and can be prone to breaking the gel matrix. The goal when inoculating plates is to streak the surface gently and not to cut, tear, or stab the gel matrix with the inoculating loop. To prepare, ensure the bench top is clean, disinfected, and organized. Make sure that tools such as loops and labeling pins are within easy reach. Laboratory personnel should use good hand hygiene, aseptic technique, and wear appropriate clothing, such as a lab coat and gloves. Ensure clothing is tight-fitting near open flames or sterilizing equipment, such as a Bunsen burner or incinerator. Allow the plate to reach room temperature prior to inoculation to avoid temperature shock to cells, which may affect recovery. Always label plates on the bottom of the dish prior to use. This is essential to identify the sample after incubation and to ensure the plate can be identified in the event the lid is lost, dropped, or accidentally switched to another plate. The purpose of the streak plate technique is to reduce the number of cells from an area of high concentration from the original sample and where the streak begins to an area of low concentration in the final streak. This is accomplished by using a series of loop passes over the agar called the 4-quadrant method. By the final streak, the number of cells on the loop should be diluted and low enough that single cells are dispersed and isolated on the agar surface. After incubation, these cells will grow into individual, pure isolated colonies. Thus, one viable cell will give rise to one single colony forming unit known as a CFU. To streak plates, 3 types of tools can be used: a non-sterile metal loop, which can be repeatedly sterilized by heat between uses, a pre-sterilized disposable plastic loop, or a sterile specimen swab. These loops come in different sizes and provide a calibrated volume, making it easier for the microbiologist to determine the number of cells per milliliter in a liquid sample. Loops are generally calibrated to deliver either 10 microliters or 1 microliter. Depending upon the testing site or sample, either a standard lab bench or biological safety cabinet, which protects the microbiologist, may be used. To sterilize a metal loop, begin by flaming or incinerating the loop. Once it grows red hot and is sterilized, allow the loop to cool in ambient air. Do not blow on or wave the loop to speed up the cooling process, and do not touch the loop to any contaminated surface prior to sampling. Cool the hot loop by touching it to a sterile area of the Petri plate. Depending upon the sample type, use the loop to collect a small number of cells from confluent growth, obtain a loop full of liquid sample, or touch the edge of a single colony from a source plate. Remove the plate lid and place it face down on the lab bench. Pick up the plate in one hand and the metal loop in the other hand. Streak the sample using a sweeping motion from side to side across quadrant 1, approximately a quarter of the agar surface. To prevent contamination, avoid streaking too close to the perimeter of the agar. The outside perimeter is where airborne contaminants are most likely to be located. Re-sterilize the loop and remember to let the loop cool before continuing to the next quadrant. You can quickly do this by stabbing the loop into the agar. Be sure to avoid the streak lines. Rotate the plate 90 degrees. Using a similar sweeping motion, streak the loop into the quadrant 1 streak lines 2 to 3 times, and then proceed to streak the remainder throughout quadrant 2, using about a quarter of the agar surface. If you used a specimen swab for quadrant 1, now switch to the metal loop for quadrants 2, 3, and 4. Only overlap quadrant 1 2 or 3 passes and no more. The idea is to continue to reduce the number of cells spread across each quadrant, so don't oversaturate the loop. Re-sterilize and cool the loop in the agar before continuing. Rotate the plate another 90 degrees and spread the cells from quadrant 2 into quadrant 3, using a similar reduction strategy and a quarter of the agar surface. Re-sterilize and cool the loop one last time before continuing. Turn the plate a final 90 degrees and streak the cells from quadrant 3 into quadrant 4. This time, sweep only once from quadrant 3 into quadrant 4 and continue the streak over the final quarter of the agar surface to disperse the cells to achieve isolated colonies. Once completed, place the lid back on the plate and place the plate in the incubator. The plate should be placed media side up, that is, the media on the top and the lid on the bottom, to prevent contamination from water condensation. An alternative to a metal loop is a plastic loop. These loops are pre-sterilized and disposable. They are a good alternative if it's not feasible for a Bunsen burner or incinerating device at the plating location. Aseptically remove a sterile loop and touch a single colony from the source plate. Never lay the loop down on the benchtop or touch it to a non-sterile surface. Proceed to streak the plate to the 4 quadrants. Use the same technique as the metal loop, only instead of heating the loop between streaks, just flip the plastic loop over to the clean side of the loop between streaking quadrants 1 and 2. Thank you for watching our 4-quadrant streak demonstration. Hardy Diagnostics is your complete microbiology supplier. Check out our full lineup of inoculating loops by clicking the link below. You can also see our complete catalog in the link below. We are 100% employee owned.
So see what we told, it looks like exactly the same. Okay. You are going to do it by yourself in hand, I guess. Yes, we used to do that. And you are going to put in the incubator. Very important. The media or plate side up and the cap down to avoid any contamination from the condensation. Okay. So exactly you see whatever they demonstrated in the video. Okay. So isolated colony, why we need that? It's very important for patient specimen with potential mixed flora. Mixed flora means different kind of microbes. Here we are not telling the exactly pathogen because we don't know. It's a flora or the microbiota. Isolating the bacterial colonies using 4-quadrant stacking is called the 4-quadrant stacking. The questions comes, okay? What type of quadrant techniques used? What is my Just give me a second dear. Everything is gone. Oh. Okay, good. So what it does, it helps to visualize the morphological characteristics of all the bacteria. Morphologicals means how it looks like, what is the shape, what is the structure, and then from the stain you can understand finally what exactly type of bacteria is there. MLTs can then test any potential pathogen using gram stain and biochemical test to identify. Gram stain is the basic one, but some tests like the TB, they are not going to stain by the gram stain. Why?
Because of the fatty wall?
Yes, waxy fatty wall. The gram stain is why it cannot penetrate. The fatty wall.
Because the gram stain is water. It has to do
Water
with
soluble, water soluble dye, so that it cannot penetrate. So what you have to use? What type of stain?
Acid
acid.
Acid
Acid.
Okay,
Okay.
acid fast stain.
Yes,
Okay, what is
acid fast stain.
the primary dye for the acid fast stain?
Carboxyl.
Carbofuchsin. Okay. What is the name? Give me name of the one of the acid fast stain which is used.
Seale-Nielsen, ma'am.
What?
Seale-Nielsen method.
Seale-Nielsen stain.
Jaden. Jaden stain. Okay.
Yeah.
Jaden stain. In short, it's called the Jaden stain. Jaden. Okay. So, and what is the counter stain?
Methylene blue?
Methylene blue.
Yes, good. And what is the
Nielsen.
decolorizer?
Acid alcohol.
Acid alcohol.
Good.
Hydrochloric acid combination.
Very good. Very good. The gram stain, what is the primary stain?
Crystal
Crystal
violet.
violet.
And counter stain,
Saffron
Saffron.
what is the color of the saffronin it gives?
Pinkish red.
pinky.
What is the color crystal violet gives?
Violet. Blue.
Bluish
Violet. Bluish.
Purple, purple or
Yeah.
purplish blue.
Purple.
Violet.
Bluish purple.
Okay. So how the crystal violet stain became attached to the bacteria?
The what's it called? It attaches to the
peptidoglycan, the structure of the
peptidoglycan,
bacteria.
of the cell wall
Mm-hmm.
Mm-hmm.
of
So
the
even
gram-positive cells.
okay,
And they possess the teichoic acid.
teichoic acid. That is
Yes.
more important. If there is no teichoic acid, even you have the peptidoglycan layer, it is not going to breach the stain. Who helps in bridging the stain with the teichoic acid?
The mordant iodine.
Iodine.
Yeah, redundant.
So even the gram, even the what is called
Gram negative.
gram negative bacteria, even they have single layer of peptidoglycan layer, but till it's not stained at all with the crystal violet because in the peptidoglycan layer they doesn't have the teichoic acid. That's why the stain cannot stay. Okay? It have to clear in mind. That is the main resource. Okay. What we are? I just go from here to there for the review. Yeah, potential pathogens using gram stains and biochemical test, and there they can do the other biochemical test to identify. The pathogenic bacteria is identified. MLT can sub out to a new agar plate to have a pure culture. So from one plate there they show you have the culture, even in the colony, right? So you can pick up one single colony, again you streak to get more isolated, more purified colony. That is the continue the running the test on. So when you are streak, you can see how it coming single individual colony. Ultimately, they are forming one individual colony. Now, even if they want for the more, they can take one, and then take streak the another one. See, isolated colonies from mixed culture. You can see the mix. There are different types of bacteria. You can see this one, this one, this type, this type, this type. So now they can pick one, do another culture. They can pick one, another culture to rule out any mixed colonies. That is more used when you need to see the antibiotic sensitivity. Urine plating purpose and techniques. Urine cultures, quantitative cultures. Quantitative urine cultures methods are required to differentiate the infection from contamination. Why? In urine, that even you are doing the clean catch urine and everything, but till there may be chances of the cross contamination because there are a lot of bacteria is in that area. Somebody didn't clean it properly, even you give the instructions. So that's why you need to do the quantitative cultures, is called the quantitative cultures, to rule out any contamination. Presence of bacteria in clean catch urine does not necessarily indicate a UTI unless the quantity of organisms is significant. Urine cultures are reported in colony forming units per milliliter of the urine, CFU per ml. It's not only the presence or absence of bacteria make it sure. When they are going to report the how many colony forming units per ml of urine. An increased number of white blood cells, that the leukocytes present in the urine specimen, which you can see in the urine analysis, and in conjunction with the results from the urine culture, is significantly more valuable for the diagnosis of infection. So you have to tally the results. Here urine analysis, oh that's why when you have UTI, doctor doesn't only order the culture and sensitivity, the routine, microscopic, RNM, and also the culture sensitivity. So if there is more leukocytes, they are all and other nitrates and other things are there when they are already changing the bacteria. So at the same time, you have more leukocytes there, and then in the urine culture also you are getting more bacteria here. So is going to tally the results. So so more leukocytes means there is an infection. At the same time, we are getting the quantitative in analysis, we are getting more pathogen here. So a central streak plate method for quantitating the growth of microorganism in urine is typically used in the clinical laboratories. So a calibrated standardized inoculating loop containing 0.000 or 0.1 ml. What is 0.0001 ml means in microliter? No Google. From your knowledge. We already passed the quality and math. 0.001 ml means how much microliter? I know you guys are doing the Google.
Microliter.
Is 1 from 1000.
Yeah, 1 from 1,000. That's milliliter. Right? Okay, now.
So what microliter? How much microliter is that? Everybody is going to get 0 now. Back to quality and math. I have to update the marks. I have to update the marks.
In air.
Everybody is going to get 0. You cannot convert 0.001 ml is equal to how much microliter?
Positive.
If it is 1 in 1000, so how much microliter?
Mu air.
What?
We're gonna check.
Nobody can answer this?
Is it 0.000001 liters?
I am asking microliter, not liter. Microliter.
It's like 10 to the minus 6.
Yeah.
For micro.
-3.
How much microliter, dear? 0.001 ml equal to how much microliter?
One.
1 microliter.
Microliter.
Ultimately Google worked, right, Bharna?
Yes, ma'am.
I know Bharna is very fast in the Googling, but don't Google. This is the
Yeah.
problem. In Google, you doesn't know how to do it. You get the ready-made answer, but if there is a different questions or different things, you cannot answer if you doesn't have the concept clear.
So, ma'am, it's like 1 times 10 to the power of minus 9.
1000 is simple. 1000 microliter is equal to 1 ml.
Okay.
Okay, now, okay, you have passed the Q and maths. I am going to take marks from everyone now. This is the very basic things. If you doesn't know how the conversion between the milliliter to microliter, microliter to milliliter, liter to milliliter, how you are going to work in the lab if you doesn't know the conversion? They are asking 0.0001 ml. You doesn't have any autopipettes which is going to write you 0.0001 ml is in the calibrations of the microliter. So you are stuck. You doesn't know which autopipette you are going to use. And you are going to ask the question. You can ask. You cannot make any mistake. But they think from where they came. They doesn't know basic things. So you have to know the basic conversions there. If it is 0.001 is equal to 1 microliter, then 0.01 ml is equal to how much microliter?
It's 10.
10? What was the
Yes.
question, Miss? I said the answer, but it was
0.01 ml is equal to how much microliter?
it's 10.
10. If it is 0.0001 microliter, how much? 0.0001 ml, how much microliter? Instead of 20, there is one more zero.
100.
Point one.
0.1 microliter. If it is 0.1 ml,
100.
100.
today's practice. Otherwise, everybody's marks will go down from the Q and math. So you are using 0.001. If it is written here, you say ml in the loop, in the packet, it says 1 microliter. You cannot understand. In the procedure is writing 0.0001 ml. Oh my God! In the inserted packet is write 1 microliter. What is that? You understand what the basic knowledge you need here? So you can use the loop volume. You have seen the loop volumes, which can contest is calibrated. So you can take only that amount, 0.001 or 1 microliter versus 10 microliter or 0.01 ml of urine to use transfer the well mixed specimen to well mixed specimens means in urine is well mixed in the container to strike the plates. So how you are going to do? This is the loops. You are going to put the loops. Then you are going to take it out. Then you are going to spread it like this. For the urine, it doesn't spread like the 4-quadrant way. That's it says. It's called the here, and this is the middle, and then you are going to stretch like this. Loop is touched to the center of the plate from which the inoculum is spread in a line across the diameter of the plate. So you are putting it here, and then without flaming or re-entering urine, loop is down cross. The first inoculum strike numerous times. So you are going from here, produce isolated colonies. Okay. So I'm going to give you the video, and can we open it?
Me again. Have you got your home insurance policy from Square One yet? Visit square1.ca and get yours in just 5 minutes.
Urine streak. The urine streak is a semi-quantitative method of determining an approximate number of bacterial cells in a fluid, such as urine. An aseptically collected specimen is taken to the lab and analyzed in the following manner. A pre-sterilized plastic calibrated inoculating loop is used. The loop is dipped into the fluid, the urine, and moved to the Petri plate. The lid of the plate is opened and a vertical streak is made down the center of the plate. The loop should be touching flat on the agar so that a maximum of the fluid is transferred. From the end of the vertical streak, streak from side to side, moving down the streak about 15 times. Follow the pattern shown on the movie. Label and seal the inoculated Petri plate. Now store the plate in an inverted position in the box or incubator for your lab session. After 24 to 48 hours, the number of colonies on the surface of the agar can be counted. The approximate number of bacteria present in the original sample can then be calculated.
Got it? How to do that urine culture? Plate stacking is not from the same like others. So in common practice, supportive media such as what type of sieve blood agar is SBA and the selective differential media can be McConkey agar extract with the urine specimen. So you can use either SBA, the sieve blood agar, or MAC, which can be used for urine stacking. Drop urine is sprayed as uniformly as possible in the plate. After stacking and incubation, the number of colonies visible in the multiplied by 1000 for 0.001 ml loop. How the calculation come, you understand? You are going to do the per ml, 0.00 ml. You have to multiply it 1000. So 1 ml 0.001 ml is equal to 1 microliter, which is 1000 times less. So for calculating, you have to multiply the pack 1000. And for 0.01 ml, which is 10 microliter, so it is 100 times less than ml. So you have to multiply it by 100 to calculate the CFU colony forming units per ml. Okay. Now is clear or no? Not yet. I asked the question. After another model, everything is gone from the head. Right? Too much
Right.
information, too much
Right.
information, too much information for
So
short time.
yeah, doesn't get the transfer to the long term, so that's why we can't remember.
Mm-hmm.
Long term, although the basic stays,
Yeah.
the
But
conversion is very good. Yes,
this
ma'am.
is very, very basic, what I asked you.
Yeah.
This is very basic. This is from the school level.
Yeah.
The calculation, between the conversion, between the milliliter to everything is a basic. This is very, very basic math. Okay. So the selective media indicates whether gram-negative bacilli are present. MAC also indicates whether the organisms are lactose positive or lactose negative. So MAC medium can help to identify both lactose positive and lactose negative of E. coli. So these questions may come: which agar plate can use to identify the lactose positive or lactose negative bacteria? You have to remember, make sure which media is using for what type of things. What is the SAB, what is the MAC, what is the SS, what is the hecton, what is the clade, whatever we have learned, which one is used for which one. So for the taking the, you can use the SIP blood or you can use the MAC. MAC helps to identify both lactose positive or lactose negative E. coli. One of the most common organisms identified in UTIs, accounts for a large percentage of UTIs in ambulatory and hospitalized patients. E. coli is a lactose fermenter. It converts the lactose. Gram-positive organisms such as Staphylococci and Enterococci grow well on blood agar. So MAC agar can be used for the Staphylococcus and Enterococci. So Staphylococcus and Enterococci can also go for on the MAC. So this is the C blood agar. Using the C blood agar, this is the MAC culture medium ultimate. This is C blood agar is the main nutrient from where they are coming, and the agar is using as a base or platform. So C blood agar, which we can make as the MAC conky. So you can use this one for Enterococci, Staphylococci, and also for the urine. Urine culture, you have seen the streak and MAC, how it spreads. Combined plates, SB and MAC. Sometimes you can get some plates where is a divider. So that in the one plate, you can do the both type of culture using the SIB blood agar and is the MAC for urine. So incubation requirement for bacteria, different environments for incubation are based on the bacterial requirements of growth. Like all, in general, the incubator temperature very important, 37 35 degree to plus minus 2. So 35 to 37. This is the base temperature or body temperature. Okay. can be an ambient air where is 20% oxygen and 3 to 5% carbon dioxide. That incubator should have the access of this ambient air temperature, where is generally you have 20% oxygen and 3 to 5% carbon dioxide. Most bacteria demonstrate enhanced growth in presence of carbon dioxide. So they favor in the carbon dioxide. So they can grow more faster. Okay, so it can create a higher percentage of CO2 environment with a candle jar. What is the candle jar? We are going to see it use a candle jar. And a nitrogen, ANA2 incubator environments are those without oxygen, which the bacteria which grows without the air or oxygen, these are called the anaerobic bacteria, right? So their environment should be, there will be no oxygen so that they can grow faster. So you have to make all these arrangements. Just give me a second. তুই আমাকে রেডি হওয়ার, তুই যখন রেডি হবি তখন থেকে বলবি, আমি তখন থেকে রেডি হয়ে বসে থাকবো, কারণ আমার তো আবার সময় লাগে, ওই জন্য বলছি। হ্যাঁ, যখন উঠবি আমি তখন বলবি, আমি তখন ব্রেক দিয়ে দেবো, বা আমি 10 মিনিট ওদের কাছ থেকে ব্রেক নিতে পারবো, ঠিক আছে? কিন্তু হ্যাঁ হ্যাঁ হ্যাঁ, তুই আমাকে তাহলে বলবি এক্স্যাক্টলি, আমি সে অনুযায়ী ওদের ব্রেকটা দেবো। ওকে, ঠিক আছে। Sorry, yeah. So these are CO2 incubator where they have the connection from the CO2 tank, where the oxygen, there will be no oxygen, mainly the carbon dioxide will be there, where the bacteria which doesn't need oxygen or cannot grow well in the oxygen. So that is the CO2 incubator, and this is called the ambient air incubator, where there have 20% oxygen and 3 to 5% carbon dioxide. And always you have to keep, you can see always that the plated things, the agar plated is going to be the face up and the cap should be down. Okay, so they are also putting different types of cultures in the bottles, in the medium, so depending upon everything. Sometimes for anaerobic bacteria who doesn't have the carbon dioxide incubator, small lab, what they does, they put candle flame, will use up the oxygen in the jar, produce an elevated level of carbon dioxide. These conditions are ideal for growth of microaerophilic organism. That means which doesn't grow in presence of oxygen, who prefers carbon dioxide. So if you burn a lamp, what is going to happen? It's going to use up all the oxygen, and when the oxygen is used up, they are going to, the candle is going to be, it's not going to be burned anymore, because candle needs oxygen, right? So that you can, and then CR side, that means only there is carbon dioxide. And when the kernel burns also, it produces more carbon dioxide. So bacterial growth requirements continued. Bacteria and other microorganisms are sensitive to oxygen concentrations. They're categorized depending on the oxygen requirement needed for growth. So obligated units, oxygen, facilitated air groups, there are different types of microaerophil, which in presence of oxygen, they cannot grow at all. So obligate aerobot require oxygen. Why? How they are differentiate the microorganisms depending on the need of oxygen to grow. Obligate, they needs, they are obligated for the oxygen. That's called the obligate aerobes. Facultative aerobots go either aerobically or in the absence of oxygen, but do better with oxygen. So this is facultative. They doesn't care. They can grow in oxygen, without oxygen, but if the oxygen is there, they grow much better. Aero tolerant anaerobes do not require oxygen but can grow in presence of oxygen. So they doesn't need oxygen for their growth. But if there is oxygen, because they doesn't need it, right? So they doesn't care. They doesn't need even per carbon what is there. Is okay. So they doesn't need the oxygen. So they can grow. But they doesn't need it. That's the one to mean. Then the obligate aerobes cannot use oxygen and cannot grow, survive in the presence of oxygen. So obligate anaerobes, okay? Obligate aerobes require oxygen 100%. Obligate anaerobes cannot use oxygen but cannot grow or survive in the presence of oxygen. Then microaerophiles use oxygen but at lower concentration that atmospheric levels around 20% grow well with higher carbon dioxide level. That is in general bacteria things. Okay. Just give me a second. I may like to make a phone call. Something is coming and I'll be back to you. Okay. Just give me a second. Sorry, there is a call from doctor's office. Okay, sorry. Okay. Second one thing. 20 people. Skara is absent today, right? Yeah, she told me
Yes,
she's going to be
ma'am.
absent. She
I
told me
didn't see
she's going
her.
to be absent. Yeah, she told me.
Yeah.
Yeah. Okay. So anaerobic cultivation in nude chamber, we have some specific.
Sorry, Ms. Manon?
Yes.
Yeah, I just want to let you know that I'm going to go to my son's ceremony in the school.
Yeah, yeah, you told
Yeah.
me. Yes. You
Yes.
are already. Yeah. So what time you're leaving?
I'm going to leave now. I might come back maybe around 12 o'clock.
Okay.
Yes.
Right. Okay.
Yeah, thank you.
No problem. Okay. So this can be done using an anaerobic chamber where oxygen remove is already done when it comes from the instrument, it comes from the manufacturer. So these containers have used hydrogen gas mixture that remove oxygen by forming water. So when there is any oxygen is there a little bit, they generally doesn't put any oxygen. But if there is any oxygen is contaminating when you are opening or closing the door, immediately the hydrogen catches the oxygen and makes it water. So there is a airlock to reduce oxygen levels during transport of specimen in and out of the chamber. So there should be not too much air is going to go inside because there is mainly the mixture of hydrogen and carbon dioxide gas. They remove the oxygen. So these are common in the lab to use create an anaerobic environment. Can be a jar box also. Compact, easy to use and less expensive than a chamber. Put your place into jar or box and open and insert a gas pack envelope. So gas pack envelope is a hydrogen which catalyzes oxygen and creates water. So put your plates into a jar or box and open and insert a gas pack envelope. What is the gas pack envelope? It's the same principle they use. They use is the hydrogen which catalyzes the oxygen present in the jar from the water. So and an indicator is placed in the container as well to visibly show the jar or box reach the anaerobic state. Close it up, incubate in regular ambient air incubator. So you can put that one inside your air incubator what you have in the lab. So this is the things. This is the you can place it. You can place it the gas pack, then close it, and then you can put inside the your air incubator. Okay, so let us video. Okay.
Brex, Dutch flower bulbs since 1818. Your best garden starts with Brex selection of flower bulbs, direct from Holland. All backed with a lifetime guarantee. Visit brexbulbs.ca.
Keep there's no voice on this one. Just sealing it so that there will be no leak. But there is no volume of this one. Our volume is total on. Anyway, you can see it. That is the gas pack. The indicator. That is the indicator, and that is the gas pack system. So you saw that one, how to do that one. There are different kinds of, in the flat one, the jar type. So all should be air sealed once you put the gas pack and the indicator. Other incubators in the microbiology lab, incubators for mycology, cultivation and fungi. So except the bacteria, we have the fungus, right? So this is the temperature in what type of incubators you need for the mycology. Microbiology is the study of fungus. Virology, study of virus. Parasitology, study of parasites. And bacteriology, obviously, study of bacteria. So mycology is the fungus. Their temperature set at 37 centigrade. Fungi grow more successfully at lower temperature. Like the bacteria prefers 35 to 37 degree, but fungi prefers, these are the questions comes, fungi prefers low temperature. Is the 30 degrees centigrade is preferable. Plates are left in for a number of days and checked daily for growth. Some fungi are slow growers. Fungi takes long time to grow than the bacteria. But incubators for Campylobacter, that is the intestinal pathogens, that's the enterococci. They need temperature 42 degrees centigrade for recovery because our stomach, you can feel it's always something is going on, eating, movement, enzyme, action, everything. So is the stomach is little bit hotter than our regular body temperature. So, and these bacteria lives there and they are used to that temperature. So they'll like to have a little bit warm temperature, which is cozy for them. So Campylobacter prefers the temperature of 42 degrees centigrade. Okay. Campylobacter. And it can be, plates can be checked after 24 and 48 hours to check any growth or, so again, they are giving the same thing, same chart and giving here what type of same slide. So this you guys can have a look and so for the urine, what type of
Man, your voice is not coming.
No, I'm not talking. I'm just looking. You can hear me now?
Mm.
Yeah, it's so small writing. I need time to see. And it's not clear also. Yes. So bacterial growth curve. The timing of incubating of our culture media is to reach the end of the log phase. What is the log phase? We are going to learn now. So we always document date and time when we start our media incubation, and then we know how long it has been in at any given time. So that's why it is very important. Your culture, you plate or inoculate any bacteria in your culture medium, you have to put the date and time along with your patient name, your initials or everything, but it's very important to put your date and time. Okay. So what is the log phase? Here we are inoculating the bacteria. This is called the lag phase. No increase in the number of living bacterial cells because you inoculate them. They are in a new environment. Like you are going to a new place, some new house. You need some time to get used to it. Where I am, where I'm getting all the resources, how I will be survive. So this is the time of the lag phase where there is basically no growth or very limited growth. Then once they feel, oh, good, I am in a good environment, I have everything to survive, then they start multiplication. Growing is multiplication means they grow in numbers. This is called the exponential phase, increase of number of living bacterial cells. This is the log phase or the exponential increase in the number of living bacterial cells. Okay, this is the log phase. Then after log phase, what happened? They have used, they have grown, they have eaten everything. They are so happy. We got everything. We don't have to do anything. We can multiply. You can use all the stationaries. But they doesn't understand. They doesn't have anything in storage because they're restricted in the plate or in some specific area. So when they used up, start getting used up, then there is a plateau in number. So they cannot grow further. So plateau in number is called the stationary phase of living bacterial cells. Rate of cell divisions and death roughly equal. So here there are no more growth. It's a stationary phase. And death of decline phase, exponential decrease in number of living bacterial cells because they are fighting each other for they are not getting too much nutrients and other things. At this phase they used up everything. So it starts slowly, bacteria is starting getting destroyed or dead. So there are four phases: lag phase, log phase, then stationary phase, and death or decline phase. Sorry, then I just give me 1 second. Sorry, just another. So sorry. So further bacterial identification of testing. So once bacteria has been isolated of the incubation period of a cultured specimen, the next step is identifying the colonies as the pathogenic or normal flora. You did that. Now you have to check whether is any infection or the normal flora. So MLT will look at the shape, color, hemolysis of the colony, depending on the type of media. Remember, some media is selective and differential and will only grow the bacteria we are looking for. The colony is suspected to be pathogenic always, and the MLT wants a quick confirmation. They can make smear on the slide, then from there they can make a smear on the slide. They can do the gram stain or specific stains to check the quality and morphology of the bacteria. And then MLT can run some biochemical tests to further identify genus and species. Say for example, they took the wound swab in the blood agar plate. So it's called the BAP is blood agar plate. MC is McConkey agar. So this you have to remember the short forms also. So there you can see what is the stacking, and ultimately they are doing this 1, 2, 3, 4, and then you can see they did the gram stain. So it's a gram stain and Cocci, right? You can see the individual one, gram positive Cocci. So how you are going to do that? It happens. Flowchart used for colony characteristics and gram stain. You doesn't have to remember so many things, but just gram positive cocci, streptococci, and staphylococci. Both are gram positive. This is, this is the gram positive. These are the gram negative. Gram positive cocci, and then you are going to divide it in this positive are ultimately is coming S aureus, micrococcus species, and S epidermis. How? Their glucose tolerance is just a staphylococcus. They are showing this one here. This is a difficult slide. You doesn't need to remember. So then this is a catalase test. How they do the catalase test? This is a control slide, and this is the negative and positive. And bacteria isolated from the blood strip agar, and they use the catalase test to check whether they have the staphylococcus species. We doesn't do that. That is the job of the medical lab technologists. So next test, mannitol, coagulase, glucose help to determine if the staphylococcus are either S. aureus, micrococcus, or epidermis. Now you know they have some glucose problem. So you have to tell which specific problem is there, which bacteria is there causing this one. So for this they need to do the additional test, which can be based on the chemical and biochemical features on the bacteria, because how is their structure, their metabolism, how they ferment. So some labs have automated system such as Malditop. I don't know this one because I don't work in the lab, but I heard that Malditop, which can identify some specific genus and species of bacteria using mass spectrophotometry. We'll talk more about this in automation. So any questions for this one? No? Okay. Anything?
Thank you.
Okay. Okay. Let me see what is the next one. Okay, this one you have this one. Already you have this one there. What is this one? Checking practice handout. Oh dear. You are going to do the practice directly on the agar plate.
Say hello to the first ready-to-use AI-powered EQMS. Our intuitive user interface enables companies to seamlessly enhance quality processes, cut costs, and deliver quality faster and more efficiently, while ensuring compliance. The .Compliance EQMS automates critical quality processes, integrating them into an all-in-one solution to help companies deliver products to market quickly and safely. .EAI was designed by AI and life sciences experts and built on industry best practices to provide ongoing up-to-date AI insights. And the industry is
What is this one?
Request a demo today to see the power of .Compliance.
Ma'am, it said.
In order to correctly treat an infection
thing,
or
right?
identify a contaminant, microbiologists must first isolate the target microbe from a sample or specimen that often contains other microorganisms. Streaking auger plates is an essential technique used in the laboratory to isolate.
Yeah, why they are repeating this one? I don't know. They give the another one again. This is the same, I guess. Ion lights, yes.
Miss, it's break time. Sorry.
No break. Just let me see what is there, okay, for the units. Checking in. So, test 2. Okay. Test 2. Just let me quickly see what is the unit 7 and unit 8. Unit 8 is the test.
Problem.
Yeah. Microbiology, parasology, superbugs. So that's how many slides are there? 49. Okay. So this we can do it. Today I am going to give you the break and then you guys can do your reviews, okay? Because you need to reviews for tomorrow's exam. Tomorrow is the exam, right?
Yes,
Yeah, so you can do that. Yeah, I can give you the time. And then after, I request you guys to after tomorrow, maybe after the test, we are going to reduce the break time a little bit tomorrow because generally you guys are going to complete the test very quickly. Okay, and then we can complete this chapter. Okay, then it's going to be easy for you guys. Is that okay? Is that sounds reasonable for you guys?
Yes, ma'am.
Yes, ma'am.
Because you need some time to review, I know. So that's why tomorrow is the test. Okay.
Yes.
Okay.
Okay.
So most of we know, but little bit we can just yeah. Okay. Okay.
Yeah.
So this is only for this one. Okay. You go for break, half an hour, and then you can come back. You do your studies. I'm going to work for something else. Okay.
No, thank you.
Amen.
Thank you,
Okay,
ma'am.
no
Thank you,
problem.
Miss.
I'll try if
Thank
I
you, ma'am.
can make some review for you guys. I am not sure, but that I'm going to try when you are going to do your first reviews. I'm going to make some reviews for you guys because from these two. Okay.
Okay, thank you, Miss.
Okay, thank you, miss.
Okay. Enjoy your break.
Thank
Okay.
you.
Okay.
Okay,
Okay.
thank you.
Okay. Okay. Everybody got so hungry with working with me. Sorry. And I truly apologize today. I hope the internet is not going to go away again. It's so depressing. In the morning, I'm clicking, and when you doesn't have the internet, I cannot communicate in the WhatsApp. I cannot. Everything is blocked.
Yeah, that's really tough. But recently, I think it's most networks have been doing that at
Right?
home. Like our Rogers has been out of control.
And not only that, I'm doing something, suddenly it's boom, out. Again, after sometimes it's again. And you have to restart everything again from there. Starting the, authenticator, these, that.
Yeah.
Yeah. Anyway, you guys enjoy your break, okay? Okay.
Thanks, ma'am.
And work for your review too. Okay, so we are going
Thank
to
you.
learn tomorrow this one. Okay, Mel. So tomorrow the exam is at what time you want? 9:15, 9:30, what time you want? 9:15 is okay?
Can we do 9:30, ma'am?
Because you have to complete after your exam, then we are going to short our break a little bit, not too much, okay? So depending upon when everybody is late from there, then we are going to short and then we are going to complete the rest.
Okay.
9:30? Okay, so we'll do
Yes,
at
ma'am.
9:30.
9:30 is
Okay,
good for.
okay, okay. Attendance. Oh, this one, attendance, attendance. Attendance. Let me do the attendance now. Microbiology. Biology, day 3, day 4. Diana present. Use of Amina present. Yasnet present. Ying Hong present. Alexia present. Zenaida present. Fasado present. Binas present. Santiago present. Joanna present. Peace present. Pada absent. Sabana present. Luis present. Evelyn present. Santiago Beato present. Maris present. Present. Close. Save. Okay. So now go to campus login. Campus login. PELACHAK number 60. Okay. কি শুনে মানে আচ্ছা ঠিক আছে আমি ওদেরকে আমি এখন রিভিউতে দিয়েছি তুই আমাকে বলবি আমার একটু
Evelyn?
Isn't it?
Yeah. You know the Stemocostus arenas and the Deocostus pneumonia, are they the same?
Now, which page is that?
Miss Mumum gave us like an handout. I'm just reading through it. She didn't put a page.
Oh, yeah. Oh, the notes.
Your notes, yeah.
What's the
Let me see what page is it. Hold on. Let me just see. 1, 2,
let's see.
4. 8.
Page 8.
9 page 9.
9, okay.
Page 9, we're in the middle of it where she lists several pathogens of men, right? Of man. So I was looking through it and I'm like, because I like to find the meaning of it.
Yeah.
While I'm reading it. And when I read, it seems like they're the same.
Yeah.
It seems like there's one name that's referred to the other, like the Stemilocostus. The other name is the Diplocostus.
Oh, the Diplococcus.
Yes.
The Diplococcus are usually in pairs. 2 pairs.
Pairs.
But what's the other one?
The stem, S-T-A-P, Steph.
Oh, Staphylococcus.
Yes.
Is this the Staphylococci or Streptococci?
Staphylococcus. S-T-A-P-H-Y-T.
Staphylococcus.
Yeah.
It's hard, eh?
Yeah, it's hard. Yeah. So I'm wondering if they're the same, but
I would say both of them are round shape, but they differ. The Diplococcus are usually in pairs only. And for the Staphylococcus, they are usually in clusters like grapes.
Grapes, okay.
Yes, but they
So
are
they're
the same in shape. They are both cocci. Like they're spherical, yeah.
Okay.
I think I have to check also. The Diplococcus are usually the Neisseria. No. Maybe I was wrong. Diphtheria? Clostridium diphtheria?
I don't know.
So they're the same family.
Yeah,
same
that's what I'm
genus.
thinking.
Yeah, same genus, but no, they differ in
colors and in shape.
Yeah, they differ in their formation, something.
But they are the same in shape. Is that what you're saying?
Yes, that's right. The same in shape. They're both cocci. Cocci is the round or spherical.
Okay, awesome. Thank you.
You're welcome. I hope I'm right.
I could ask Miss Mum because she lists them. She lists them as a pathogen of man. And those are the two, and there's Salmonella. That's the other, it's the third one. So I like to find the meaning to understand it.
Yes. Yeah.
Okay. Awesome. Thank you.
You're welcome. What I remember is the Staphylococci species. They are in a grape-like clusters. They are the same.
That is a gram-positive. Huh?
Yeah,
Sorry.
they're the same like the Streptococci, but the Streptococci are they are elongated, but they are also connected like in chains. But still the same shape. They're still round shape.
Rounded shape.
Right.
Yeah.
That's right. You're
Okay.
welcome.
Awesome. Thank you.
Welcome.
Someone? I'm back. What are we doing now?
We're just reviewing our notes, like we're studying.
Oh, okay.
She gave us the free period to basically study.
Oh, study the review, you mean?
Yes, study the review and like the
Okay.
slides. Yeah,
Oh, okay. Okay, got it. Thank you. Thank you, Christian.
no problem.
Hello, guys. Is there anyone who'd like to start the review? We have less than 30 minutes to go. Is there anyone who would like to read it? I think Evelyn might need a break at this point. So is there anyone else who'd like to start it? Okay. So I guess not. So I guess not. Okay. Take care.
So sorry, Asenath. It's just I'm not fully done the review yet.
Yeah, mine too. I just still in page.
I'm on like page 9 right now.
I'm almost done.
I'm still working on it as well, but if you guys want to do it later in the evening or something, I don't mind. We can go through it for like if anybody has an hour, we all can
I
join.
wouldn't
and
mind
then-
either.
Or we can do after class.
Oh yeah, or after
Maybe.
class too.
Yeah.
Yeah, let me just as soon as I'm finished, then I will just let you guys know. I'll drop a message in the group, and if everyone's ready by then maybe we can start reviewing it, and I can read the questions, no problem.
Okay, that sounds good.
Okay, perfect.
Yeah. Good idea, Asnet and Shabana.
Thanks. Good luck, guys.
Yes, you too.
Okay, guys. Thanks.
Pinapokay, saglit. Saan? Ay, naririnig mo ako. Ay, oh shoot.
How is the review going? Good?
Yes, ma'am. Still working on it.
Good. Okay. Good. Drumstick. Good. Paste. Paste 52 glycan. 52. কি হলো? হ্যাঁ বল। অলমোস্ট শেষ হয়ে যাবে কেন? Carbonyl fusion. Carbonyl fusion. It is called Jidenstin. Okay. Jidenstin. Yes, suffering. 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 Groupstin. Distant themselves. Okay, Ramstein. Still violet. I accept. Yes. So it's one. Class time is over. If you want to stay in team, you are welcome. If you want to do by yourself, whatever, you guys are welcome. You guys are allowed to leave now, as per the things. Okay. Okay.
Thank you, Miss.
Okay,
Miss.
thank you.
Yes.
Thank you, ma'am. Thank you.
Thank you, ma'am.
Yes. Okay.
Miss, can I talk to you later, please? I just
Yeah,
have a
sure.
question. Thank
You
you.
can, yeah, you can just do it just after, because I'm going to go to work, so you can do it just after the class is over. You can call me, okay?
All
Sure.
right. Thank you, Miss. I appreciate that.
Okay,
Thanks.
okay, okay. Bye, everyone. Wish you good luck for everyone, okay?
Bye.
Thank you.
Okay, okay.
Thank you,
Thank you,
Miss.
ma'am.
Bye,
Bye.
bye, bye. If you want, you guys can have a group meeting in the evening if you think is useful for you guys. Whatever wants to join. Sometimes I saw the group reviews gives you better results instead of the individual one.
Yes.
Yeah. So it's up to you guys how you, if you want to have it, not it is, it's ball is in your court. But I tried to send you the review, work on that, and hopefully everything will be okay. Good tomorrow. Okay.
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 Thank you,
Thank you.
Miss.
Okay. 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 Bye now. I'm leaving. Okay.
Thank you,
Okay.
Okay,
Miss.
ma'am. Bye.
Okay.
Bye.
Bye.
Thank you.
You guys are ready for the review?
Yes.
Right now, I don't mind, but I will have to join you guys by around 2-ish or 1:30 because I have to go pick up my kid first. Is that okay?
So should we do it in the evening or are we doing it
If you
now?
guys want to continue, go ahead because I already did most of my questions, and I'm confident they're good. If not, I'll just join. You guys can continue first.
I'm okay with anything. It's whatever the majority prefers. If you guys want
Yeah,
to do
or
it
unless you
now.
guys want to take a break right now, do your thing, and then we
Or
can
another thing
meet
I
in the
could do
evening.
is we could do it now. I can screen record it and then I can email the screen recording to whoever wants it for further review. So
Yeah, we could
Shabana
do
would
that
be,
too.
we could do that as well. Whatever the majority prefers.
Sure, I don't mind anything. I'm open for anything, guys. Honestly, whatever everyone is comfortable with, I'm good with that. But it'd be good if you can record it because I feel like Kara is in here and like a few people who are not here, they can also get the reference and they can follow up if they want to.
Yeah, okay, sure. Yeah, okay. So if we want to get it over with now, hands up to whoever wants to do the review now. Just, on Teams. I wouldn't mind. I'm either or.
Yeah, I mean, I still have like 15 minutes. I can do the reading first and like continue, and then when I have to leave, then somebody can take over.
Yeah, okay.
Is that
All right.
okay?
Yeah, sure. So do we want to do that? Do we want to start now then? Let's get like a yes
Sure,
from
we can start now. I have
All
my,
right,
let me
cool.
just
Let's
get
start now. Who wants to lead?
I'll start. I don't mind starting.
Okay, yeah, sure. Gee.
Okay. For
Um,
does everybody have their stuff opened? Yes,
Give me one
Vinesh,
second.
can I help you?
Yeah.
So, guys, do we start now?
Yeah, we're just starting now. I'll lead the class for a bit, and then after that, someone can take over because I have to go somewhere. Okay? But we're gonna start now with the review if you guys are ready.
Okay.
Let's go ahead,
Yeah.
Okay.
Shabana. Yep.
Yeah, I think we're
Okay.
good. Let's start.
Okay, guys. So I'm gonna go with the first question first. Which of the following statement is accurate for blood culture specimen collection? I chose number 1.
Oh, I chose
Uh,
number 3.
You
Mine is
chose number
number 3 too.
Yeah,
Yours is
3.
number 3. Okay. But when I Googled it, I mean that's what I got. But okay, number 3, right?
Yep.
Everybody
3.
agrees with number 3?
Yep.
Okay, perfect. Number 3 it is. What is the function of transport media? Mine was to sustain pathogenic bacteria from testing. That's
3.
number 3.
Yep.
Okay. Okay. Sputum C and S has been ordered by a clinician. What is not one of the best practices for collecting this specimen? I went with number 1. Patient should wait until
Yep.
they have a sneeze
Number
and
1.
then expel.
I
Yeah.
got that too. Yep.
Okay. Okay, going forward, disinfect your work area with 10% or
Yes.
1/10 bleach solution after each lab session. That's what I wrote. Okay,
Yep.
what is the best time to collect a blood culture set? Mine is number 2, prior to
2.
administration of
Yes.
antibiotics.
Yep.
Why is activated charcoal used in some transport medium in swabs? I chose number 4.
4. Yep.
Yeah.
Yep. Okay. When setting up the color illumination on a microscope using a 10x objective, what is the total magnification of the image? I did number 2, which 100 times.
100 times.
Yes.
Yep.
Okay. In the lab, you can use chapstick. I said false. You cannot use anything from outside, and because it's going directly on your mouth. You are working in the lab with gloves. Lab phone rings and no one is available to pick the call. So what will you do? Mine is number 3, remove gloves and pick up the
Mine.
call.
Yes. Number 3.
Yeah, me too. Number
Okay.
3.
You noticed your colleague is chewing gums.
Gums.
Do you think they
Nope.
can? So I said no.
Nope.
Okay.
No.
Prokaryotes genetic material is DNA.
Yeah.
Yep.
That's the
Cellular
answer, right?
DNA. Yep.
Yeah, that's what I wrote. Color illumination acts to generate / illumination of the even. I put number 4, even.
Yep.
Yeah.
Okay. Low individual and community risk falls under risk group 1,
1. Yep.
Yes.
right? And then the staphylococcal species falls under select all that apply. So I selected 3, which was risk group 1, moderate risk group, and risk group 3.
Oh, it's 2 and 4. Moderate
Yeah.
risk
4?
group and risk group 2.
Okay. 2
Yeah.
and 4,
Yeah. I
right?
got 2 and 4.
Okay, perfect. I updated that. Okay, risk group 4 is considered number 2, high individual risk and high community risk,
Yes. Number 2.
right? Okay. Ebola virus is categorized under risk group number 4. That's what I wrote. Okay, what is a role and responsibility of the MLA/T with regards to microbiology specimen collections? I went with number 3. The MLA/T supervises all patient collections to ensure accuracy.
I chose number 1. I'm
Oh,
not too
mine
sure.
is number 1.
I also
Yes, number
chose
1.
number 1 for that
Number
one. A.
1.
Yes, number 1.
Number 1? Okay.
Yeah.
Yeah.
Perfect. No worries. Okay, continuing. Okay, so this one you guys want to go through one by one. Like for MRSA, the first one I put bacteria.
Uh-huh.
Okay, number 2, COVID-19 virus. Number
Virus.
3, malaria is parasite.
Yeah.
Number 4, HIV is virus.
Virus.
I can't even pronounce this, but it's bacteria.
Yeah.
Yeah, it's bacteria.
Influenza, I put virus.
Yeah.
Tapeworm, I put parasite.
Parasites. Yeah.
Candida SPP, I put fungi.
Mm-hmm.
Beaver fever is a parasite. Asparagus SP mold.
Fungi.
Fungi, which
Hepatitis
mold
B,
basically.
Yeah.
Fungi and
virus.
molds. Yeah.
Yeah.
Yes.
And then hepatitis B is virus.
Mhm.
Yeast is fungi.
Mhm.
Mumps is virus. Mushrooms is fungi. Rubella is a virus.
Oh, wait.
Pinworm.
Viral infection. Yes, virus. Sorry.
Yeah, viral infection, right? Pinworm is parasite. Chickenpox is a virus. Rust is a fungi. And measles is a virus.
Yes.
Yeah.
Okay, so everybody got that?
Yes.
Perfect. Okay, what is the order of draw when blood cultures are ordered? I put number 1, serum tubes, anticoagulant tubes, blood culture bottles.
Wasn't it B?
And number 2.
A 2.
Yeah,
Oh,
blood
sorry,
cultures
yes.
go first.
Yeah.
Blood culture
cultures
bottles,
first,
serum
and
tubes,
then
and
serum, and
anticoagulants.
then anticoagulant.
Yes, number 2. Okay, number 3, we are going, virus can survive without a host. I put false, it cannot.
False.
Yes.
Yeah.
Okay. The next one is choose the right order. For me, is number 2. Is it correct?
Yes.
I wasn't so sure.
Yes.
Yeah, I think
Two.
so.
Kingdom, phylum,
Yes.
class, order, family, genus,
Yeah,
and then
yeah,
species,
that's
right?
what it is, yeah.
Okay.
Okay. I got that one wrong.
No problem. You can put that down. What is the reading on the red needle at BSC safety gauge? I put 0.51.
Isn't it 0.55?
Mine
yeah,
is 0.55
because it goes on increments of
0.55.
5.
because
Yeah.
the other line is
Okay.
0.75 when you're counting like 5 by 5.
Yeah.
I don't know.
Oh,
Yeah.
okay, okay.
And
If
you
you
just
look at
put
it,
the
it's
number.
like it's counted almost like by fives, like
Mhm.
50,
Yeah.
55, 60, 65,
Right.
70, and then 75.
And you want to read
Okay.
it to like the line that is closest at. So like you wouldn't want to put 5.54 or 0.54. You want to put 0.55.
Yeah.
Fair enough. That makes sense. Yeah. Okay. Moving on would be destruction of an microorganism, including bacterial spores, is called sterilization.
Yep.
Yeah.
Right?
Yes.
Okay. What is the advantage to working in a biological safety cabinet in the microbiology lab during a setup? I put number 3. It protects both the specimen and the lab worker.
Yes.
Yep.
Okay. So next is bacteria can survive without a host. I put.
Yes.
Right? Okay. A hot air oven needs 160 degrees to destroy spore forming bacteria. That's what I put.
Yeah.
Yeah.
Is that correct? Okay.
Yeah.
What is the best definition of normal flora? I put number 1, microorganisms living on a host without causing harm.
Yes, I got that too.
Okay. The process of destroying pathogenic organisms, accepting the spores in terms of, is a term as in disinfection,
Yes.
Yeah.
right? Okay. The containment level requires HEPA filter. HEPA filters is containment level 3.
Yes.
Okay, a biological agent which is potentially hazardous to a life is called pathogen,
A pathogen. Yes.
right? What
Yep.
term is used to describe an infection acquired by an individual while they are patient in the hospital? I put number 1, opportunistic infection.
It's
Oh,
nosocomial
it's number
infection.
4.
Is
Four.
which one? Number 4?
Yeah.
Yeah.
Nosocomial. Okay.
Nosocomial infection. Yeah.
4 it is then. Okay. All of the following are patient instructions to collect feces for ONP, except avoid contact with the SAF preservative fluids.
I put number 3.
Yeah,
Feces cannot be
yeah.
taken from the toilet. I don't know if that's
I
correct.
also put
Yeah, that's
number
my answer too.
Yeah.
But I'm
Okay.
just saying, like, does that mean, like, is it, like, what if, how do we know if they're not doing that at home, though?
I
I mean,
know.
I'm sure the results will be
Right? That's what I
skewed.
meant, though. That's what I'm for number 3.
Yeah.
You know,
But
but
I got
anyways,
number
okay.
3. I don't know.
Okay, we're for number 3. What? But I feel like I read it somewhere. It says like contact with the SAF, like they cannot like get in contact with the SAF preservative fluid because it's a preservative fluid. If they touch it, it's not preserved anymore, right?
No, it's just it's used as an agent to preserve the sample itself. The reason you wouldn't want to take it from the toilet is because there's bacteria of built
Yeah,
up feces and
exactly.
urine that have been chilling there for ages. So,
Yes.
that would skew with the results, and I'm sure that MLT would notice
Yeah.
in the
Yeah.
results.
And there's
But
also
I
like
feel,
cleaning agents
yeah,
in
but I
toilets
put
too.
4 because I feel like if they get in contact with the SAF preservative
It could be
fluid,
dangerous.
it
I
will
do see that.
contaminate the preservative, right?
I do see why, like, coming in contact with the preservative could be dangerous, like if you have an open wound or something. But essentially, if it's liquid, you just wash it off.
Fair
But
enough.
yeah, I think C is the best
Yeah.
answer in this
Number
case because
3.
I don't think anyone's gonna like dip their finger in the preservative fluid anyway.
Exactly.
You can
Hope
tell
so
them in
not.
advance.
According to D2L, there
According
is
to
already a contamination with the
the
bacteria
toilet
in the
bowl,
toilet.
yeah.
Cannot
Okay.
for the
Okay, so
culture.
number 3, right?
Yes.
Yeah.
Yes.
Okay, number 3 it is. What container is used to collect a CSF specimen for C and S? I put number 3, plane 2. Is
Yep.
it correct? Okay.
Yes.
What is the term for a person who cannot fight a pathogen that has entered their body? I put susceptible
Yes.
host, which was number
Yes.
3.
Susceptible host. Yes.
Okay. Select the vector-borne pathogen. I put number 2, West Nile and dengue.
Yep,
Yeah.
that's what I got.
Which statement is false regarding viruses? I put number 3. Most viruses are easily destroyed with chemicals. So I put number 3 because I think most are not easily
My
destroyed.
answer
with
here is
chemicals.
2.
I put 2. Viral diseases
put
are
2?
treated with antibiotics. Yeah.
Yeah. 2.
2.
Yeah, I also put 2. Yeah.
It's 2.
Number 2. Okay. Number 2
It
is.
is 2.
Perfect. Okay. Carrie Blair transferred media preserves the viability of bacterial. I put enteric pathogens.
Yep.
Yeah, enteric.
That's what I
Yes.
got.
Okay. Which statement best describes the
Yeah,
characteristics
enteric
of
or intestinal. According to
Sorry?
D2L, enteric or intestinal.
Okay,
Or
enteric or
intestinal.
intestinal. Okay.
Okay.
So
Enteric
both.
or intestinal. Okay.
Yeah.
Thank you.
Perfect.
Thank you.
Sorry, guys. Well, which questions?
We are on
Uh,
page 5, question 6 on page 5.
Yeah,
I know they're
page
not
5.
numbered. Yeah, page
Carrie-
5, the 6th question on page 5.
Yes.
So the Carrie Blair one, I think.
Oh yeah, the Carrie Browse there. Yeah,
Yeah, Carrie Blair. We're on
what's the
number
answer?
6.
Actually, you know what? I don't know why I'm not sharing my screen with you guys. I should just do that.
Yeah, the Carrie
Right.
Berry transport media.
I'll just share this. Okay, can everybody see my screen?
Yes.
Yeah.
Okay. I think this will be easy. Can everybody see all the wording? Is it clear enough? Or should I make it larger?
Oh, that's good. It's clear.
It's good?
Yeah. Yeah.
Okay,
Shivana, that's
perfect.
good. Thank you.
Okay, and this way everybody
That's
will know which
awesome.
question we're at.
Yeah.
Okay. Anyways, we're over here, guys. Okay.
Mhm.
Okay, carrier player transference. So that's the enteric or essential pathogen. And then the next question: which statement best describes the characteristics of a prokaryotic cell? I put number 2, like you guys can see.
Yeah.
Is
Number
it correct?
2.
I got
Yes.
that.
Yes.
Okay,
Yes.
perfect. The next one is vector-borne transmission refers to infections caused by
Birds and rats.
arthropods and
Arthropods
insects.
and insects.
Yeah,
Sorry, what are the
arthropods
two?
and
What are the
insects.
2 answers for that? I wasn't too sure myself. What are the
Me
2
too.
vectors?
I put it over here. You can see it's the
I got like
arthropods and
insects and
insects.
animals.
Animals and
That's what I
insects.
got.
That's what I got. Insects and
Animals and
animals.
insect. Yeah.
I
Insects
got
and animals.
arthropods too.
So
I put
Arthropods and
arthropods as
mosquitoes
well.
and ticks.
Yeah. Okay, I'll
Yes.
ask Miss Moon about this in case though.
Yeah. I think it's insects
Um,
and animals. I'm pretty sure.
that would make
insects and animals.
sense. I mean, that's pretty
Yes,
broad. So yeah.
that's right.
Yeah.
Because animals like the pig, we get the tinea from that.
Yep.
Like
Fair.
birds.
Animal and insect.
Animals
Yeah.
and insects. Mosquitoes are considered as insects.
Yeah.
Or the
That's
ticks are insects too.
okay. So I'll just put over here animals as well. Okay. And insects. Which of the following bacterial names is allowing proper nomenclature rules? I put number 1.
It's number 3.
Oh, I
My
got
answer is
number
number 3.
3.
You guys put number 3?
Gonorrhea.
Yeah.
Yeah.
It's number
Okay.
3.
Perfect. No worries. Okay. Liquid or particle suspended in gaseous medium is an aerosol?
Yeah.
Yep.
Yes.
Okay. What specimen is submitted to find intestinal parasites? I put feces.
Yep.
Yes.
Okay. What safety equipment is used while planting patient specimens directly to media? I put number 3, biological safety cabinet.
Yes.
Yep.
Okay. What are the parameters for steam autoclave to ensure sterility? I put 121 and it's 15 psi in 15 minutes, which is number
Yep.
3.
Yeah.
Okay. I didn't do this, guys, so you guys can help me out. The first one, identify the picture.
So the
It's
first one is
Yeah.
cocci.
coccus.
The first one is
Cocci.
coccus.
The second one is spirillum, and the third one.
Cocky.
Is streptococci.
So the
And
balls
the
are
The
coccus.
third one is staphylococci.
Yeah.
Okay, so the
Okay.
first
I
one
don't
is MON.
Okay, let me type it in chat.
Wait, what's the last one?
just spell it out. I'll
Staphylococci.
type it here so everyone can see it.
Okay,
Sure. It's
MON.
Cocci. Yeah.
COCCI.
Yeah.
US, right? Or is it I?
That's a singular. Cocci is plural. Yeah.
Yeah.
It's gonna be
So
okay.
cocci because there's so many. It's
So
plural.
Okay, cocci.
But there's more than one Verna, so it'll be caucus, right? Like it'll be
No,
C
cocci.
Cockeye?
Cocci is the
plural.
Cocci.
plural.
Coccus is singular.
Yeah,
Cocci
Okay.
is plural.
Okay.
Okay, and the string thingy.
Spirillum.
Okay,
I
SP.
don't know. My answer there is streptococci. I'm not sure for
Yes,
everyone.
Yeah, that's what
you're
I got.
correct, Berna. Streptococci number
It's
2.
STRE
There's like
Yeah,
three
they are in
different
chains.
names.
Strepto.
Yeah,
S-T-R-E
streptococci.
P.
E P. Is it S-T-R-E-E-P?
No,
No,
only
No.
one.
STREP.
One E.
Singly.
Like strep throat.
P now. P.
P. T-O.
T-O.
Cocci.
One word.
Strepto.
Yeah,
Cocci.
one
You
One
word.
have
word,
to put
Shabana.
T-O. Yeah.
Okay, one word. Okay.
O.
T-O. You have to
There's
put
O.
O.
Strepto.
S-T-R-E-P-O?
Yes, T-O.
Okay.
Yeah.
P. Is this it? There you go.
Yeah.
Yeah.
Yes.
Okay. I didn't know I had that. Anyways, the next one, the last one.
Staphylococci.
Yeah. Verna is
Streptococci.
correct too.
Yeah.
There's like
Staphylococci.
three different names for these guys.
S-T-A.
Star
Alpha.
S-T.
Apple,
S-T-A-P
yeah. P.
P-H.
H
Y.
Y.
L-O.
L-O
Cocci.
C-O-C-C-I. Yeah.
Yeah.
Right.
Okay, perfect. Okay, everybody got that? Okay, moving on. Which term best describes the process of destroying pathogenic organisms, but possibly not all microorganisms or endospores? I put disinfection.
Yep.
Yeah.
Okay, specimens collected for ONP is done using the formalin fixative container.
My answer here is SAF. I don't know. I searched in the Google.
Yes.
It's
Yes.
SAF.
SAF.
S-A-F.
SAF is more, yeah, SAF is
Okay.
more appropriate.
So SAS, you said?
No.
SAF.
SAF.
Yeah.
SAF.
S-A-F. Like
S-A-F.
Yes.
okay. Urine is normally sterile within the bladder, true?
Yes, true.
Okay.
Yes.
Steatorrhea is too much fat in the feces,
Yeah.
right?
Yeah.
Okay.
Fat,
Excess fat.
Cystitis is an infection of the bladder, and then fungi causes ringworm infection in humans and animals.
And this one I have no clue.
Yeah, that's what I read too.
And next one, like you guys can see, I didn't really put much, guys.
B-A-C.
Bacilli.
Bacilli.
Yeah. I-L-L.
C-I-L.
I.
I.
Okay.
Okay.
Yeah, that's correct.
And the next
And
one,
this one is what?
Vibrio.
Vibrio.
B-I.
B-R.
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 Vibrio.
No.
No, B as in boy.
B as in Bob. No. V-I-B.
Oh, V-I-B. V-I-B.
R-I-O.
Yeah.
R-I-O. Okay, Vibrio.
Vibrio. Those are so hard to pronounce, guys.
I know. I hate pronouncing it.
Remember.
Makes me look like a fool. Okay, guys.
The
Next one is
S-P.
S-P. S-P.
I-R.
I-R.
O.
O.
C-H.
C-H.
E-T-E.
E-T-E. Okay, perfect. Okay, guys. Can everybody see it clearly?
Yeah.
Yeah.
Okay,
Then
next one is, what is this one?
Next one.
Spirillum.
Spirilla. S-P.
S-P.
I-R. I-L-L-A.
Okay, perfect. Everybody got it?
Yeah.
Okay. Identify the false statement regarding microbiology specimen collection. I put a 24-hour specimen can be used.
Yep.
Yeah.
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 When skin scraping is received in the lab, what organism will be testing it for? Usually for fungi?
Yeah.
I put number 4. Okay, specimens for microbiology should be obtained before number 1, administration of antibiotics.
Yeah.
Yep.
Okay. To obtain urine samples for CNS, instruct the patient to cleanse the perineum with a cleansing towelette from back to front.
Front to back.
I have front to
Front to
back.
back. Sorry, yeah, front to back, actually.
Yeah.
That makes sense. Oh, yeah, front to back it is. Okay.
Yeah, Miss Momo
And
mentioned
then,
that too.
Yeah. A dwelling which is fully placed directly into bladder and secured there for a duration of a time.
My answer here is extended period of time. I'm not sure for everyone.
Okay, so
Yeah, period. Period. The blank is period of time.
period of
Oh,
time.
period.
Yes, I have period of time.
Period of time. Okay. Which of the following is present in the collection container for O and P? I believe it's number 3, but correct me if you can. I put number 3.
Yes.
Possible 2, sodium acetate also.
Yeah.
Number 1, you mean?
Yeah, possible 2. But I have a question mark for the formaldehyde because it says the formaldehyde is a lower concentration of the formalin.
Yeah.
That's true. I
We
guess
can
that's
ask her
why.
this one
So,
because it's
I'm just
very
gonna put
yeah,
both
so we
confusing.
in
don't
case, and then we
know.
can ask her. I'll just highlight both
Yeah.
though for now.
Yeah.
Okay. Okay. Specimen injection criteria for C and S urines. Select all that apply. I only left number 3 out, and I selected all the rest.
Yeah, that's what I got.
1, 2, 4. Right.
Okay. 1, 2, and 4. Okay. In class 1 cabinet, um, air is HEPA filter, then discharged to the outside environment.
Yeah, outside.
Yeah, atmosphere.
Okay.
Yes.
Or atmosphere.
Okay, atmosphere outside. Okay, perfect. Documentation for all specimens sent to the microbiology laboratory should also include the source, such as wound, sputum, and body fluid. I put true.
Yeah, I have true.
Yeah, true.
Okay. UV germicidal lamps produce UV lights at 254. Is that
Yeah.
correct?
Yeah,
That's what I got.
Okay.
nanometer.
Nanometer wavelength. Okay. Which of the following classification schemes for all living organisms can be described as having the closest relationship? I put number 4.
I was
Isn't it-
confused about this one. I have species and then
I
I
have
thought it was number
species too.
4.
Isn't
Yeah.
it
Number 4.
species?
I
What is
Number
it
I
for?
don't know. I saw it
Closest
somewhere. I thought it
relationship.
was number 4.
So
Yeah, the genus.
It
Genus,
is?
right?
Yeah.
Okay.
I don't know. That's what I got, and I saw it from one of the notes or something. I remember that.
Yeah. Okay. Then
Yeah,
it's number 4.
genus.
Yeah.
Okay. Thank you.
Okay. HEPA filters removes particles from air,
Yes.
or
remove.
what is it?
Yes.
Removes,
Yeah.
right?
Remove.
Removed.
Okay.
Yes.
Yeah.
Yeah.
Okay. HEPA stands for
So I thought it's biological particles from air.
biological. Okay.
So it removes biological particle.
Yeah.
Is
It
this high
filters
efficiency
the
particulate
biological.
air?
I'm pretty sure that's it.
Filters biological or
Says
it removes?
filters.
Filters
So what does
biological?
yeah. My answer is biological.
Filters biological particles from.
Oh, okay.
Okay.
But it could be
biological.
anything.
Yeah, I have removes, but
I have
bio-
removes too.
Yeah, mine too. Me too.
Okay. The next one is HEPA stands for high efficiency particulate air.
Yep.
Yes.
Okay. What is mycology, the study of? It's fungi.
Okay.
Yeah.
Yep.
Yeah.
Which of the following is preferred specimen for urine culture and sensitivity for adults? I put early morning CCMSU, which is number
Yes.
3. Okay, UV lights can be left on while anyone is in the area, which I put as false.
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 Okay, in a compound microscope, the total magnification observed is the product of the magnification of objective and eyepiece lenses,
Yeah.
right? Objective. So the
yes.
answer is objectives and then eyepiece.
Eyepiece.
Yes.
Okay.
Yeah.
If hands are visibly soiled, you can sanitize using sanitizer. False, you cannot. Okay. Stage is the horizontal platform on which the slide is placed to observe the object. I put stage. I don't know what to use.
Yeah.
Okay. The usual magnification of an ocular is 10x.
Yep.
10 times basically. Okay, the following you guys can see bacteria is parasitology. I put parasitology as C, which is parasites. Number 2, mycology over here is D, which is fungi. And then I put bacteriology is A for bacteria. And then, of course, the last one is B for viruses, okay, for virology.
Yeah.
Okay. Next one is resolution indicates how small and how close individual objects are, like dots can be and still be recognizable. So I put
Yes.
resolution. Is that
That's
correct?
right. Yeah.
Yes.
Okay. Which controls the amount of light passing through the material under observation? I put iris diaphragm.
Yep.
Yes.
Okay. Number one, common type of illumination for general laboratory work is bright field.
Bright field.
Yes.
Yeah.
Okay. Which nomenclature is correct? I put number two.
Yeah, too.
Two? Okay.
Yeah.
Oh, I don't know why I didn't do this one. This is a
I
weird
don't have
cycle of
number two.
infection.
Okay, I have to change mine. Okay. I have number four.
The next one I didn't put anything, guys. I don't know why I
My
left this out
answer
though.
for that is number
It's
3.
number four.
Agent
Yes, number three.
reservoir. Yeah.
That's what
Okay.
I
Number
have.
three.
Three.
Yes.
Three.
Okay, guys, perfect. Okay, treponina is the pathogen for syphilis.
Yeah.
Yes.
Yes,
Okay.
that's right.
Other name for nosocomial infection is the hospital-acquired infection.
Yeah.
Yeah.
Yes. Or
HAI.
HAI.
Okay, which stands for hospital. Yeah. Lyme disease is by whatever you could. I don't know. Yep. Bacteria belongs to prokaryotes.
Yeah.
I put this
All
one.
right.
Prokaryote.
Yeah.
Okay. Bacteria are pathogens.
Microorganisms.
Oh no,
Oh,
microorganisms.
microorganisms. Why did I put
Yeah.
pathogens? I don't
Yeah.
even know. I
C.
think I highlighted it wrongly. It's like what? Okay. A microorganism that causes disease is a pathogen.
Yeah.
Yes.
Okay. Okay. Escherichia coli normally resides in the number 3 intestinal tract.
Yeah.
Yeah.
Okay.
Yeah.
Cocci is round in shape.
Yeah.
Okay.
Yeah.
Staphylococci are arranged in pairs, are arranged in chains, so 2 of the above.
I have a question in here. Is
Yes.
Staphylococci is it's the answer here. The D is the arranged in chain is the
Arranged in chains
streptococci.
is strep. Yeah.
Streptococci, yeah.
Yeah.
So 2 of the above is
Arranged
correct.
in pairs. Yeah.
Arranged
The
in pairs, yeah.
diplococci
But
also.
see, like I feel like
Diplococci.
Miss Munmun already like I didn't highlight this, you know. It was
She
already
highlight that.
highlighted.
She highlighted
Yeah,
it.
so that's why I put 2 of the above.
Yeah.
I think she like
I
kind of
have
gave
a
us the
question
answer.
for this. I
But
don't know.
no.
This is
Staphylococci they're
wrong.
talking about is the question is about staphylococci only.
Yeah.
And
Yeah.
Isn't it also
staphylococci
fast
are
producer?
in clusters.
Right.
They are
Yeah,
in
so arranged in
clusters,
pairs
in
and arranged
grape-like
in chains,
clusters.
right?
Yeah, so my
Okay, so
answer
that's
here
why
is
she
B,
highlighted it.
B
Okay.
are past producers.
fast producers.
I think
Because
so, yeah.
Thank you. I
because if
have B.
because if they're talking about cocci, then yes, A and D are correct.
Yeah,
Yeah.
right.
And you're
They
correct, right?
can be
Okay, but she
in
already
chains,
highlighted
they can
that for
also
us
be in
though.
pairs. Yeah, but my answer
Okay.
here is B. I'm going
Yep,
to
B.
it's
go for
B. It
B.
was a mistake on her part, I
Fast
think.
producers, yeah.
Okay.
Mm-hmm.
The past producers, right? This one?
Yes.
Yeah.
They are past producers.
Okay. And then D is not right?
Yeah.
Yeah,
D
I
and
will
A
go
are not right, I
for
don't
B.
think.
Yeah.
Okay.
Because when I was
Alrighty.
researching, it didn't make sense.
Yeah, fair
Yeah,
enough.
because of the staphylococci. If we're talking about the cocci, then yeah, makes sense. A and D.
Yes.
Yeah.
And B also.
Okay. Okay. Staphylococcus aureus causes everything except I put balls, wound infection, and meningitis absence instead of UTI. I think the
No,
only thing that
UTI
doesn't
is
cause
the
is UTI,
answer.
right?
UTI
Okay.
is it?
UTI.
Yeah, so
UTI.
it
Letter E,
causes.
except
Except.
UTI.
Yeah.
That's right.
Yeah,
That's what
but I highlighted because I wanted to know. So don't go to
Yeah.
mine. This is my reading to remember.
Oh,
But
okay.
yeah, it just doesn't cause UTI. Some
Yeah.
bacteria form a hard protective covering resistant to heat and chemicals called a spore.
Yes.
Yeah, spore.
Okay.
Yeah.
Okay, bacilli may be none of the above.
Spore former.
Is the
Spore
spore
Yeah.
former?
former.
C.
C.
Maybe spore
C.
former. Letter C.
Yeah.
Okay. Alrighty. Moving on would be cylindrical shaped bacteria that have a lot of flexibility and movements are spiral. First one.
Yeah,
Letter
A.
Okay. On a microscope, what adjusts the amount of light coming into the lens system? I put the diaphragm. Number D. I mean, well, it says DD, but it's actually E.
E, yeah.
Yeah. What is the total magnification of the oil immersion objectives and 10 times ocular? I put E.
Yes.
1000x.
Right. Okay. Which objective allows the largest field of vision? I put scanning object.
Yeah.
Yeah.
A.
Okay. Or
No,
oil
really?
immersion, which is
I have no
oil
idea
immersion?
about this. I have a
My
question
ans-
mark about
yeah, my
low
answer
power.
here is low power objective.
Yeah,
But I'm not
Oh.
so sure. Yes.
low
I
power
have-
Okay, let's
objective is
ask
my
Miss Minmin
answer.
Let's
about
ask
I
her.
this.
have a
I have
question
A.
mark.
Yeah, so okay, I'll just
Question
put a
mark,
question
okay.
mark over here. So we can just ask later.
Mm-hmm.
Okay, next one, antiseptic blood culture called blood culture is iodine and chlorhexidine,
Hexidine. Yeah.
Yeah.
right? Okay.
Yeah.
The bacteria on a stain smear are observed with light microscope or oil under oil immersion.
Yes.
Light,
Compound
yes.
light microscope if you
Compound
see
light, right?
pathology. Yeah.
Compound, okay.
Compound light microscope.
Compound light. Oh, okay. COMPOUND. Okay. What is the morphology of spiral? Spiral. I put spiral shaped or corkscrew shaped.
Helical or spiral shape?
Helical.
Yeah,
Yeah,
I
that's
got
what I have.
Helical
Yeah,
or spiral shape?
helical.
coiled. Yeah.
Okay, so spiral or what was it again?
Helical.
Helical.
H-E, oh, or yeah,
Heli
CL?
Cally.
Yeah.
Yeah, that's correct.
Okay.
And then
Perfect.
dash.
Midan,
Coiled.
Well, yeah,
Put a dash and then coiled.
Sure.
Okay. Dash and then coiled. COILED.
Yeah.
Yes.
Okay.
Yeah. And the
Guys,
That's it.
Yeah,
oh
we're
wow,
done
okay.
now.
I did make it. I was quite quick.
Yay.
Okay. It took longer to do than to
What did
actually
you
just
guys
read it out.
What did you guys get for number one? I wasn't in here what did you guys get?
Three.
This is what I
3.
put. Number 3,
Three?
not number. Yeah, not this one. This is wrong. Don't follow mine. This is correct.
3.
This one.
Number
Yeah,
3
this
is
one is.
right?
Yes,
My
bottles are
our
filled
answer
until the
is
vacuum is.
A.
My hand says
Mine is A too. That's what I put, but then I thought we agreed that it was an A.
Oh.
Both
Should we put a
aerobes
question mark?
and anaerobes.
Yeah.
Yeah,
Mine
well,
is
that's what I put earlier, but I wasn't sure. But then
Okay, let's ask
everybody
her.
agreed on number 3, but I
Yeah,
put
let's
number
ask her.
1.
I got
Okay.
3.
Yeah,
Question
that's
mark
why
there.
actually, you know what? That's why I think I highlighted it twice because I wanted to ask her.
According to D2L, the volume of blood that's supposedly in the culture media should be 10 only. So
Yeah.
it won't be filled until the vacuum is reached. So there's a
Okay.
10 ml only, maximum. There's a line where the blood should be put on.
Yeah, so number 3
10
is not
ml.
possible, right?
So this
Okay.
is one.
Yeah.
So it's not.
Okay, that makes sense, Evelyn.
Yeah, it's not like the prothrombin time that until the vacuum is complete, like it will start on its own.
Okay, so
Okay,
after the
so it's A.
question,
Sorry, number 1.
the other question with the disinfectant your area, I see you have 10% there. Isn't that supposed to be 1?
No, 1/10 bleach, right? The bleach that we make at our like when we are working in the lab, it's how we make the bleach.
1% bleach.
Yeah, so 10% to depending on the amount of bleach you're making. So if you're making, say what?
Yeah.
1 to
Always
9.
1%.
It's 1 to 9, so it's 1%.
Is it 1 to 9? I thought it was always 1 to 10, right?
No, 10 is the
dilution factor.
Yeah, the total.
So it's going to be 1 to 9.
Yeah.
The ratio is 1:9.
1:9.
1 to 9.
That's the ratio.
Okay.
For the dilution factor is 1:10.
Yeah,
Okay. Okay.
that's true.
Mm-mm.
Okay,
Yes.
that's it, guys. Are we done?
What about the other question? Why is let me see. Why is it activated charcoal?
This one over here? Yeah.
Number
Why is
yeah.
activated charcoal used in some transfer medium in swabs? I
Yeah.
put to absorb toxic substances.
Not 3?
Maintain a neutral pH because most of the bacteria are neutral to maintain it.
Okay. I thought it's to like if it has a leakage because charcoal has an absorption factor. But okay, I'll put it. I'll put number 3. Because I did some research on it. For some reason, they said to absorb the toxic substances.
It doesn't
Which
absorb
one?
because the word here is absorb.
Hmm. Okay. I will move this. So this one
Mine
is
here
number
is
3,
4.
right?
I don't know.
Yeah, mine was 4 as well.
4.
4?
Yeah, let's
I'll
Because
just put I'll put
Mostly
both
someone
for
uses a
now.
charcoal to put
Let's
inside the refrigerator, right? To absorb the odor. That's what I'm thinking.
I know, but
I
you know
don't
what?
know.
I'll just put over
At this, yeah.
here.
yeah.
I'll just put a note next to it.
yeah.
We'll just put
Okay.
question
No,
mark.
Asinet will be asking Ma'am Moon.
no, let them go.
No.
No, just type it and send it to her. Because maybe she's sleeping right now. So
No, she's going to work.
maybe you should just
I mean, we can just ask her tomorrow before we start because she comes in at
Yeah.
9. We'll just ask her
Right.
the 3 questions.
No. Send it to her in our chat, and
Yeah, in
maybe
the
when
chat.
she's at work,
Yeah, in
Okay.
WhatsApp. Actually, in WhatsApp we can do that.
Oh, that's true. In the WhatsApp, yeah.
Yeah.
Okay.
Yeah,
So we
send it
know
to
before
in the
tomorrow.
WhatsApp.
Because she said
Yeah,
she's not working tonight.
Okay.
yeah.
Fair
That's
She
enough.
why I
said
don't
she's
bother
going to
to
work
call
right now,
her.
though.
Yeah,
Yeah.
she's
Sorry,
not.
guys. Please
No
don't send
Yeah.
it to our WhatsApp.
WhatsApp.
don't do
Internet,
WhatsApp.
Okay. Okay, guys, I need to cut, so I'm gonna stop sharing for now.
Oh, yes. You have to
Okay.
go pick up your son. Okay.
Shabana,
Yes.
yeah,
Yes.
Shabana, can you just scroll down? I just want to make sure, quick take a picture
I
about
wanted-
the multiple question fungal virus. That page.
Which page?
Scroll down.
Up over here?
Down. Yeah, down.
Down here?
Yeah, down. No, down. It's
More?
multiple fungal. That's that one. Yeah.
Okay.
Okay. Yeah, I just take picture. That's it.
No
Okay,
problem.
got it.
Go ahead.
Yeah, that's
Got
okay.
it?
Thank you.
Okay,
Yeah.
perfect.
Number 4. Let me see one question on number 4. Number 4, the last one, two, the
This
third
one?
question down. The contamination of the HEP. Number 4. Yeah, go down. Contamination. Go down.
This
Maybe.
one? Oh no, that's
Where is
containment.
it? Contamination. I don't see it yet. Number 4, contamination of the H. I don't see
Read
it.
the whole question.
The contamination level requires HEPA filtration is contamination level of. It's on number 4.
Stage
I'm on my
4?
page number 5. This is number 4. This is my page number 4. See?
Okay, it's after the process of destroying the pathogenic organization.
Go lower, Shabana.
Go lower. No, go down.
Containment.
There.
Asinet is talking about the containment.
This
Yeah.
one? The containment
Yeah.
filter. Yeah. Okay, level 3.
It's level 3?
Yes.
Yeah.
Okay, because I wasn't so sure of that one.
Thank
Me
you.
too.
Okay.
Thank you. Thank you.
Thank you,
lady.
Shabana.
Okay,
No
so
worries.
Shabana. Thanks.
All right,
gonna
you guys.
set
Take
the-
care. All the best, guys.
Yes, you
Thank
too.
Yeah.
you. Take care.
Take
Take
care.
care.
Bye.
Okay, bye.
Bye, guys.
Bye.
Thank
Bye,
you.
everyone.
Bye.
Good
Bye.
night.
Bye.